This video demonstrates our imposting framework for accelerating the rendering of quadric elements such as spheres, cylinders, and helices for visualization of large biomolecules. We implement our framework in Proteinverse, a tool for visualizing various representations of large biomolecules using standard desktop graphics hardware. Proteinverse has been developed at Visualization and Graphics Lab, Indian Institute of Science, Bangalore. The Spacescale model is a common representation of primary structure of molecules where atoms are represented as spheres with radii set to their van der Waals radius. In our framework, we first render a rectangular impostor that covers the sphere's rasterization. Each raster fragment is then checked to see if it belongs to the sphere. If it does not, it is discarded. The retained fragments are depth corrected and colored using accurate surface normals. This allows accurate rendering of spheres at any resolution. Here we render the GROYEL or 1AON molecule, which consists of approximately 60,000 atoms. Proteinverse uses our framework for this purpose and achieves a frame rate of about 55 to 60 frames per second. The details of the card are as shown. The Ballstick model is another common representation of the primary structure of molecules where atoms are represented as uniformly sized spheres and bonds between them as cylinders. Similar to the spherical impostor, the cylinders are first approximated by a bounding cuboid and the impostor's retained fragments are depth corrected and colored with accurate normals. The spheres are then rendered using the spherical impostor primitive. Proteins consist of a contiguous chain of amino acids referred to as the backbone. They are commonly represented by sampling line segments from a spline curve passing through the alpha carbon atoms of each of the amino acids. The cylinder impostor method demonstrated earlier is used for each line segment to produce a tube-like object. The impostor is modified so that there are no gaps between continuous segments. Surface normals are interpolated consistently across consecutive cylindrical segments to ensure smooth shading. Oftentimes, weak hydrogen bonds form across amino acids in the backbone chain of proteins, leading to formation of secondary structures such as alpha helices and beta sheets. The alpha helix commonly exhibits high structural regularity, with the helix executing a turn for every 3.6 amino acids in the backbone and a pitch of 5.4 angstroms. Also, the coiling is regular across a standard axis. We leverage on this regularity to fit a cylindrical impostor across this axis that is appropriately culled to reveal the helical structure. In conclusion, we have presented our framework to accelerate the rendering of large molecules by augmenting the OpenGL pipeline with spherical, cylindrical, and helical elements. We applied our methods to accelerate various representations of primary and secondary protein structures such as spacefill, ballstick, backbone loop, and alpha helices. By using fewer primitives to represent the structure, we utilize lesser graphics memory and can scale our framework to large biomolecules while maintaining high quality and interactive frame rates. We conclude the video by giving a quantitative comparison of PyMol against Proteinverse. The specs of the molecule being rendered and the card are as listed.